Molecular evolution, the concept of neutral evolution. In 1968, Moto Kimura proposed the neutral theory of molecular evolution. The Kimura suggests that at the molecular level, the majority of the mutation are selectively neutral. That is, they confer neither an advantage nor an disadvantages on their barrier. Thus, the majority of evolution changes in macromolecules and much of the genetic variation within the species could result neither from directional selection of advantageous alleles nor from stabilizing selection, but rather from genetic drift. The neutral theory of molecular evolution states that the vast majority of evolutionary changes at the molecular level is caused by the random drift of a selective neutral mutant. The theory was received by some as an argument against Darwin's theory of evolution by natural selection. The Kimura maintained, and most evolutionary biologists agree that the two theories are compatible. The theory does not deny the role of natural selection in determining the course of adaptive evolution. Kiruma in 1986. However, the theory attributes a large role to the genetic drift. According to the Kimura, when one compares the genomes of existing species, the vast majority of the molecular differences are selectively neutral. That is, the molecular changes represented by these differences do not influence the fitness of the individual organism. As a result, the theory regards these genomic features as neither subject to nor explicable by the natural selection. This view is based in part on the degeneration genetic code, in which the sequences of three nucleotide codons may differ and yet encodes the same amino acids. Example, GCC and GCA both encodes alanine. Molecular divergence and molecular clocks. The molecular clock is a technique in molecular evolution that uses fossil constraints and rate of molecular changes to deduce the time in geological history when two species or other taxa diverged. It is used to estimate the time of occurrence of events called speciation or radiation. The molecular data used for such a calculation is usually nucleotide sequence for DNA or amino acid sequence for protein. It is sometimes called as a Jane clock or evolutionary clock. The molecular clock, some proteins seems to change or evolve at a consistent rate. The number of amino acid constitutions in the homologous protein is proportional to the time elapsed since two species diverged. The DNA comparison also have been used as a molecular clock to date phylogenetic branching. The data obtained by this method are generally consistent with the fossil records. The consistent rate of protein change and the rate of DNA divergence in the group of related species indicates that the accumulation of the neutral mutation may change the genome as a whole more than do changes brought about by the natural selection. The disagreement about the extent of a neutral mutation has caused some evolutionists to rely on the molecular clocks to determine the sequence of branches in a phylogeny, but not the actual dates of origin taxa. Classification and identification. The goals of systematic is that the classification reflects the evolutionary relationship between the species. Each taxon should be monophyletic, containing only species that are derived from a single ancestor. A polyphyletic taxon includes groups having different ancestors, and a paraphyletic taxon excluded some species that shares a common ancestor with other species in the taxon. After diversion into unicellular plants and animals, the complexity increases slowly. Consequently, the diploblastic animals evolved, and then triploblastic coelomates and coelomates. One of the bases of similar characteristics, animals and plants are grouped into phylum, class, and order. Molecular tools in phylogeny. In the history of molecular phylogenetics study, the data suggested that the DNA sequences may indeed evolve and diverge at a constant rate, which is certainly not true of morphological features. This concept has been dubbed the molecular clock. Zucker-Candle and Pauling in 1965. To the extent that a precise molecular evolutionary clock exists, it can provide a simple way of estimating phylogeny. It can also enable us to estimate the absolute time, since different taxa diverge. If we can determine how fast it is ticking. Less complex and more complex prokaryotes, all eukaryotes, algae, protozoa, fungi, vascular plants, arthropod, chordates, fish, amphibians, birds, mammals, and reptiles. The genomic size variation. The bar indicates the range of genome size for particular clades. The taxa are arranged top to bottom in order of increasing the organismal complexity. Restriction mapping. The restriction mapping of DNA compares the fragment of DNA from different species cut by the restriction enzymes. If two species have diverged greatly, their collections of restriction fragments will not match closely. The restriction mapping of mitochondrial DNA, mtDNA, which mutates about 10 times faster than the nuclear DNA, provides the phylogenetic information for closely related species or population. Examples of restriction mapping: 15 kb DNA fragments on enzyme digestion to form enzyme A restriction fragment, enzyme B restriction fragment, and enzyme A plus B restriction fragment. Enzyme A restriction fragment 2.5 kilobase, and enzyme B restriction fragment 5 kilobase, and enzyme A plus B restriction fragment 2.5 kilobase and 5 kilobase. On conclusion, the enzyme A outs 2.5 kilobase from one end of the fragment. The enzyme B outs 5 kilobase from either the same end or perhaps the other end. The A plus B shows that the enzyme digests at the opposite end of the 15 kilobase fragment. Therefore, it forms 2.5 A to form 2.5 kilobase and B to form 5 kilobase. DNA sequencing. The DNA sequencing determining the actual order of a nucleotide in the DNA, providing the most accurate information on the genetic divergence between the species. The sequencing or ribosomal RNA whose gene change slowly can be used to determine early phylogenetic relationship. The analysis of fossil DNA molecular systematics has now been applied to DNA extracted from fossils. The small quantities of DNA are amplified using the polymerase chain reaction and tested from the homology and with the modern species. The ancestral RAG1 to RAG2 transposon insert to form ancestral receptor genes. Then the split gene to form ancestral split receptor genes. Then transfer of RAG1 and RAG2 gene from split gene to new place in the genome to form RAG1 and RAG2 gene becomes functionally independent. Then in excision and recombination activity to form a matured antibody mRNA rearranged by RAG1 and RAG2. Then the second round of transposition yield modern split receptor genes to form then on gene duplication diversified immune receptor gene repertory to form a matured antibody mRNA rearranged by RAG1 and RAG2. The protein product of RAG1 and RAG2 performed the recombination reaction of a vertebrate immune system by binding to a recombination signal sequence RSS adjacent to the receptor gene coding elements. The key evolutionary events gene coding elements. The key evolutionary events were TOP, the initial insertion of an ancestral RAG containing transposon into a progenitor variable region receptor genes, thereby splitting it into different elements with the RSS flanking coding region, the ancestral split receptor genes. The subsequent to this transposition, RAG1 and 2 genes are lost from the split gene and becomes functionally and genomically independent. Protein and nucleotide sequence analysis. Although the biochemistry of organisms are not well known in Darwin's time, the modern biochemistry indicates there is a biochemical similarity in all living cells. For example, the same mechanisms for trapping and transforming energy and for building proteins from amino acids are nearly identical in almost all living cells. The DNA and RNA are the mechanisms for inheritance and gene activity in all living organisms. The structure of genetic code is almost identical in all living things. This uniformity in biochemical organization underlies the diversity of living and points to evolutionary relationship. Cytochrome C. It is the respiratory pigment found in all eukaryotic cells, which is a part of electron transport system. The cytochrome C accept H plus in all eukaryotic cells. The cytochrome C molecules of human and chimpanzee is identical, whereas neurospora differs from the human's cytochrome C by the position of 44-104 amino acids. Two blood groups, A, B, ABO type of blood groups are found in human, whereas apes have only A and B types, and monkey lacks such type of blood groups. This indicates that the humans are more closer to apes than monkeys. 3. Blood and lymph. The composition and the physiological role of blood, lymph and tissue fluid are similar in all vertebrates, which shows evolutionary relationship among all vertebrate animals. Hemoglobin is a conjugated protein having two alpha chains, each with 141 amino acids in man, and two beta chains, each with 146 amino acids in man. There is a single amino acid difference between human and gorilla hemoglobin chain, and 26 amino acids among human and horses, whereas 10 amino acids between human and pig. The DNA comparison, genome comparison is a direct measure of shared ancestry. The DNA hybridization is a technique that measures the degree of base pairing between the single strands of DNA from two species. The more extensive the pairing, the tighter the hybridization strands are bound, and the higher the temperature needed to separate them, indicating a greater homology between the DNA sequences and a closer phylogenetic relationship. The evidence from this technique has been used to settle some old taxonomic disputes. The protein evolution and species divergence. 200 years the birds from reptiles, and after 200 years after mammals from reptiles, fibrinoplasmid. Then after 400 years, since years species divergence millions of years, reptiles from fishes, and 600 millions of years vertebrate from insects. Hemoglobin is formed. Fibrinoplasmid were birds from reptiles, mammals from reptiles from fishes, and 600 million years vertebrate from insects. Hemoglobin is formed, and cytochrome C. Typical function of AFGP gene from an Antarctic nototinoid, showing the repeated THR alanine motifs encoded in the exon 2 that comprises the antifreeze domain. B. A typical trypsinogen gene showing the single tyrosine THR alanine motif at the beginning of exon, as well as exon 1 and 6 in red that is found in the AFGP gene. C. The chimeric AFGP gene from a giant Antarctic toothfish, Dissostrichus mawsoni. The exon in blue encoded the THR alanine repeats is a many-fold expansion of the single motif located just upstream of the exon 2 in the trypsinogen gene. Origin of new gene and proteins. The new gene origination is a driving force of evolutionary innovations in all organisms. The recent research has focused on identifying the mechanisms that generates new genes, and scientists have found that these mechanisms involves a variety of molecular events, all of which must occur in the germline to be inherited by the next generation. After the germline mutation event, the new gene examples a new gene duplicate located on the human chromosome 2, which will be polymorphic in the population. In other words, not all second chromosomes in the population will carry the duplication. The last step necessary for the evolution of life was the origin of genetic information. The successful protobiont would need only to go and divide, but also to develop a mechanism for replicating the successful characteristics, for creating instructions for making their key molecules. One hypothesis maintains that the first gene were short strands of RNA, which have been shown in the laboratory to be capable of self-replication. The Czech recent discover of Czech's recent discovery of RNA catalyst called as a ribozyme indicates that RNA molecules may have been self-replicating in the prebiotic world. Early population of RNA molecules may have undergone the natural selection as those molecules with the most stable three conformations and the greatest autocatalytic activity within a particular environment successfully competes for the monomers and generated family of the similar sequences. The RNA-directed protein synthesis may have begun with the weak binding of specific amino acids to the bases along RNA molecules and the linkages to form a short polypeptide. This polypeptide may have then behaved as an enzyme to help the RNA molecules replicate. Mechanism of new gene generation. This includes the gene duplication, transposable elements, proteins, domestications, lateral gene transfer, gene fusion, and gene fission, and de novo origination. Gene duplication. The gene duplication was first mechanism of gene generation to be suggested. Ohono 1970, and this process does not indeed appears to be the most common way of creating the new genes. Transposable element protein domestications. The transposable elements TES or so-called selfish segment of DNA that encodes proteins that allows these segments to copy or move themselves within the genome. There are two types of transposable elements: DNA transposon and retrotransposons. 2. The lateral gene transfer. The scientists use the term lateral gene transfer to refer to the case in which a gene does not have a vertical origin, that is, the direct inheritance from parent to offspring, but instead comes from an unrelated genome. It is well known that this sort of transfer occurs between the bacteria, and that is also has taken place between the genome of cellular organelles, mitochondria and chloroplast, and the nuclear genomes. Roger 1999. 4. The gene fusion and fission. Existing gene can also fuse, that is, two or more gene can become part of a same transcript or undergo fission, that is, a single transcript can break into two or more separate transcripts, thereby forming a new gene. Interestingly, it has been observed that the chimeric fusion gene sometimes involves two copies of the same gene. Examples: the alcohol dehydrogenase gene in Drosophila. 5. De novo gene origination. New genes can additionally originate de novo from non-coding regions of DNA. Indeed, several novel genes derived from the non-coding DNA have recently been described in Drosophila. Began et al. In 2007, Levin et al. In 2006. Gene duplication and divergence. The gene duplication appears to be the most important process that enables the proteins to acquire new functions. The gene duplication is any duplication of the region of DNA. It may occur as an error in homologous recombination, a transposition event, or duplication of an entire chromosome. A second copy of the gene is often free from the selective pressure, that is, mutation of it have no deleterious effect to its host organisms. When a gene is duplicated, one copy of the gene is potentially freed from having to perform its original function. The copy is redundant if the original gene is still producing the original protein. Therefore, the gene duplication may allow the evolution of entirely novel protein functions without impairing the cell functioning. The gene duplication may involve a part of genes, a single gene, a portion of chromosome, an entire chromosomes, or the whole gene. Duplication of entire genome polyploid have been important in speciation, especially in plant. Autoploid individuals are usually viable because all of these chromosomes are duplicate, thus they avoid imbalances in gene expression. The plants are most prolific genome duplicators. For example, the wheat is hexaploidy, a kind of polyploidy, meaning that it has six copies of its genome.